viewing because apparently I talk slowly, and so you can watch it on YouTube at double speed, and then it's sort of a quicker pace to get through all the material. Um, okay, so to kind of start off, I'll do a little bit about sort of, who are the people who are teaching the course, and so not all of them are on there yet, and it will probably change a little bit, but sort of what's our background, sort of who are you and what's sort of expected. And so we've gone around a little bit and covered a bit on your background, and we'll get into sort of more detail on that. And sort of why does this class exist? I mean, if you look through the ETH course catalog, there are lots of image, vision, computer science classes available. Where does this try to fit in? And then a bit, what's going to come up with the rest of the lectures and what are sort of the core important concepts of this class. And then we'll sort of get into, what's an image, where do images come from, science and reproducibility. So this is more about kind of the higher level concepts of, why is it important to do image analysis well, and what does reproducibility mean? And then this idea of workflows. And so the tool I sort of mentioned before, KNIME, is a workflow tool. And so why we use this and how this mindset is very important even when you're doing Python or Java or C++ or whatever other languages you might be using, that having a clear model for this will help make it easier to do your work well and easier for other people to understand what you did, take parts of what you did and apply it to other things. And of course, if you have any questions, feel free to raise your hand and ask them at any point. It's not interrupting and it makes it much more interesting. So in terms of who I am, so I founded a startup 4 years ago to do image analysis. So we worked quite closely with hospitals in the medical image analysis area. And there we were really focused on machine learning to automatically identify cancer and tumors. And we have a few example data sets in the exercises later if you're interested in that sort of particular problem. But we also worked on other areas with satellite images and similar sorts of projects where we saw a lot of what's being done today in image analysis and a lot of sort of the big challenges. I was formerly a postdoc at the X-ray microscopy group. I think one student's from there, where we were doing analysis of 3D tomography images. And so what made them even more interesting was the fact that you had time evolution as well. So you could do really fast imaging and you could actually watch structures change in 3D. But of course, this makes a ton of data because each image, I think, was 14 gigabytes. And so if you had thousands of images, you ended up with terabytes of data that you needed to analyze. And then, yeah, formerly a PhD student at the Swiss Light Source at Paul Scherrer Institute. The TA for the class, which will come for the exercises, is Amoga. He's also from this group and is working on sort of experiments in similar fields and is quite experienced with both Python and KNIME, and so should be able to answer most of your questions, and works in this building 2 or 3 days a week. So if you have questions, finding time to meet up with him shouldn't be a problem. Then one sort of the guest lecture for the third lecture, which is quite important, is from Sync, which is the synchrotron source at Paul Scherrer Institute. And so they have really, really noisy, ugly images. And so he's the one who has to write all the tools to make those images usable for other things. And so has put together really nice lecture material for how you clean and enhance images well and sort of what things you need to think about when doing that. And then one of the other guest lectures that we'll have is from the Nexus Personalized Health sort of Technology Center at ETH. And he was formerly at Roche doing high content screening. And so I think there were a number of students who had examples or had issues similar to this, where you basically are able to screen lots of cells and you have machines that can do millions of experiments per day, but you need a way to analyze all of these images quickly and reliably in order to figure out which drugs might work and sort of move on to the next candidates. So in terms of who you are, sort of typically there's a very wide spectrum. I think there was only one computer science student. Was there more? Only one, okay. So yeah, so from the sort of high end of computer science students where you've done lots and lots of programming, you're very comfortable with vision, information theory, those topics, to art history researchers, where we had an art history professor take the class because they were analyzing old paintings and they were interested in sort of quantifying the different layers. And they weren't really interested in writing tons of code or understanding how compilers work or anything like that. And so the class is really meant to address a fairly broad audience where it doesn't expect you to have a huge amount of mathematical background or programming experience. And so sort of the wide range of skills from sort of I've heard of MATLAB before to I write optimized C++ code and everything else. And so if you are sort of in this group, you'll have opportunities to do things that are sort of more complicated and go into exercises and problems that are more difficult. But if you're more in sort of this group, you'll have the ability to at least finish all of the exercises and understand a little bit about what those next steps would be for if you need them or if you're working with groups where you need to have that. So kind of how will this ever work? It seems like quite a difficult starting point. So again, this conceptual graphical assignments with practical examples. And so really the emphasis in this course is what steps do you apply in what order and why do you apply these steps? And sort of, why is it better to use this kind of filter instead of that kind of filter, instead of how do you implement this and how do you make it efficient and everything else? And so really the focus is understanding how all of these different pieces fit together and to put them together in a reasonable way. And then if you know a lot more and want to do custom things or special high performance operations, you'll have the ability to do that, but you won't be forced to do that. So in terms of course expectations, for exercises, there's usually one set per lecture. The exercises sort of aren't mandatory, but strongly recommended. And this is then really much more for your own benefit, that a lot of these concepts are a little bit abstract and trying to understand them is a lot easier if you have a real problem that you're trying to solve instead of just, looking at a slide and saying, okay, that makes sense. And, the easy ones will be sort of using GUIs, so KNIME and ImageJ, connecting the right components together, sometimes completing scripts. So actually we fixed that lecture now, so it's all Python based. And then the more advanced ones will be writing more of your own code to solve more complicated problems. But really the emphasis is strongly on how do you apply these different concepts together. Then the second part, which I actually think is one of the most important parts of the class, is this idea of a science project. And so this is also optional. The only thing that's required to get credit for the course is to take the oral exam at the end. But what this is basically taking the concepts we've learned in this course and applying it to a problem you care about. So obviously for the PhD students, this is quite a bit easier because you have data, you need to analyze it anyways. And doing a science project as part of this class gives you a structure for analyzing this data. And then at the end, everyone will present and you can get feedback and tips and insights on sort of what, how you could maybe solve your problem better or different approaches you could take to certain parts that are difficult. And it gives you the ability and everyone the ability to see sort of what people are using in this course and which tools work well and which ones don't. And so I find this part actually quite valuable for most students because, the exercises or data sets that I have or I've found, and this is then really your own data where you care about what the results are and you want to analyze it better. And so I think that's quite useful. And we'll get into that in more detail, I think, in the second lecture. So in terms of literature and useful references, each slide set or each presentation has this generally in it. There isn't really an official textbook for this course, so you won't need to buy one. All the links are available at ETH, and so if you're interested, you can click into it. Sometimes it's kind of helpful to have some of these books and go through them, but they're not essential. So each lecture will have different ones that they reference. I find the first sort of two to be very helpful if you're just looking to have a book to go through, but the ones specifically named in the lectures will probably be more useful to that material itself. There's also all the old material from the courses, and I link to a number of other courses on the course website. So if you go other materials at the bottom, and this list is ever expanding. And so, if you're interested in specific or more complicated tools, you can always find it there. So in terms of kind of today's material, so for the imaging, there's a nice presentation on sort of ImageJ and scientific Java, which is sort of the basis for what KNIME does in image analysis, which is sort of the tool we'll go over today in the exercises. A bit about cloud computing, although we won't talk about that as much. And then this idea of reproducibility. So we have a lecture that's really dedicated to this later, sort of particularly with the statistics, but trying to come up with a basis for why reproducibility is important and why we make so much effort for going through this workflow and everything else in the class. So in terms of motivation, the kind of broad motivation for the course is this diagram here. And so that you have kind of, a bone, for example, you take an image of it, you do all of these sort of magic steps in between, and then you have this article that says, sort of science says ice cream is good for bones. And really the point of this course is to try to disentangle all of these things and understand how to do these things correctly so that you can come to this conclusion. I mean, not this conclusion specifically, but that you have the ability to take a bunch of image data and turn it into a scientific paper, or at least a complete, somewhat coherent thought about what this data means. And so it's trying to take apart, what image processing steps should you be doing? What statistics steps should you be doing? What do you need to be measuring? What do you need to be focused on? How do you know you're not making sort of simple mistakes in what you're doing? And the second part of this, which is really where the big component comes in, is how do you go from one analysis on one image to 100, 1,000, 10,000, 1,000,000 without making more work for yourself? And so really, how do you make all of this very, very scalable? Because I guess as most of you who've worked with images before, if you're doing anything manually, it then takes a huge amount of time to do large numbers of samples. And so how can you do this much more efficiently? The other sort of motivation for this course is in kind of the detectors are getting bigger and faster, and today's detectors are very quick. So at one of the beam lines at the Paul Scherrer Institute, they have a detector that's capable of measuring 8 gigabytes of data a second. And so, on this laptop, for example, the memory would be filled in 2 seconds. And so you need new kinds of approaches to analyze this data in a reasonable way so that you can handle the amount of information you're collecting and you can get results in a reasonable amount of time. And we see that for this camera, it's more information per day than Facebook and 3 times as many images per second as Instagram. And so it's really on the scale of the largest, most successful companies. And this is just one camera at one scientific institute. And Facebook and Instagram have thousands of computer scientists and servers and experts. And so you have the same data challenge, but a lot fewer resources. And how do you do that efficiently? So these are just a few examples from sort of X-ray and optical. I think some of these might be a little bit out of date, but for diffraction patterns, they have a detector which can do 30 gigabytes a second. At other beamlines, the data rate isn't quite as high, but you still end up with very, very large data sets. And so if you do a day of measurement at the nanoscopium beamline in France, then you can end up with 10 terabytes of data. And so this is obviously a very unwieldy amount to analyze. And even optical experiments, you can end up in situations where you get 500 megabytes a second. For the newest GoPro, you can get 60 megabytes uncompressed, and I think over a gigabyte a second compressed. 60 megabytes compressed and over a gigabyte a second uncompressed of data just from recording a GoPro continuously. And so the second part of the motivation is if you look at sort of the breakdown of experiments in a very broad way, so that you have experimental design, management, measurements, post-processing as sort of the four different steps of doing an experiment, that's shifted a lot. So that if you look from the year sort of 2000, you had a little bit of experimental design, and then you had quite a bit of time taking measurements, then managing, and then doing the post-processing. That was a fairly balanced process, so that, not a quarter of the time for each step, but at least the division was fairly clear. And that if you look as sort of time has changed, the measurements have gotten faster and faster and faster. And so you can see sort of 2014, they're almost invisible. In 2020, they're gone. And so that basically, at least at some of the experiment facilities, we've gone to a point where you can take 3 seconds worth of data and spend a whole PhD analyzing it. And so the ratio between how much time you need to spend measuring and how much time you need to spend analyzing has shifted massively. And so if you look at 2000, all of these skills were sort of equally important. And if you look at today, it's much more important to be good at data analysis than it is at measurement, because the measurement's over in 3 seconds and that's already done. And so this is really a response to that shift. And how do you make scientists who are dealing with these images every day much more comfortable with how you analyze it and aware of what tools are available? So in terms of how much is a terabyte really, this number gets thrown around a lot, and it's a little difficult to actually see. If you take sort of an image, and this is then sort of the first code that we'll have in the course, if you take an image like this, which is just 1,000 by 1,000, and you were able to look at one of these images every second. So you looked through that, and maybe this is cells or something, and you're trying to count how many cells. If you were to look at one of those images every second, it would take you 138 hours to go through a terabyte of data. And so it's just wildly beyond your ability to actually look at each image individually. And so with anything like this, you'll need automatic analysis that's capable of quantifying the things that you're interested in, counting the cells, finding different objects, identifying features that could be of interest, identifying bad measurements. All of those things aren't things you can do manually, because it would just take you way too long to get anything done. So if we look at kind of this process, if we take, this image, for example, if you were to count how many cells were in this slice, you wouldn't need code to do that. You could just manually go through and count all of the cells. But if then you had the challenge of trying to count, are there more cells on the left side or the right side, you'd have to start over again and count everything over. And then if I told you, these long stripy things aren't cells, and probably these circle things aren't cells either, you'd have to start over and count again. And so all of these sort of minor tweaks cause you to have to redo your analysis completely. And sort of as most of you, I guess, have PhD advisors or have worked on research projects before, the first task that you get is rarely the last one. And you very frequently end up with, every single time you come up with an answer to your advisor's question, he has 3 more questions that he wants you to answer that require you to restart and do everything again from the beginning. And so the faster you can do this analysis and make it flexible, the easier your life is going to be. If you were to do this again with 96 samples and 2,000 slices instead of just 1 slice, of course, this then gets well beyond your ability to analyze it in a reasonable way. And then if you do it again for 1,090 samples, each one of them has different cells, different structures in it, and you want to start comparing what are the different shapes mean and how are these related to each other, then you get into a very, very difficult problem. And so you're way beyond anything you can really analyze easily yourself. And so where it really gets better, as some of you also mentioned sort of at the beginning, was when you have sort of softer qualitative metrics. And so if you're looking at cells in bones, for example, and you're trying to describe how well aligned they are. You have kind of aligned, well aligned, not really aligned at all. And so maybe this works well for 3 samples. But what happens when you have thousands of samples? How are you supposed to go through and rank how well aligned each one is? You'd have to go and take each sort of pair of images and decide which one was more aligned, and then try to come up with sort of a ranking for them. But it would be very biased, very arbitrary, and if you ask someone else to do the same experiment, they would potentially get different results. Is this really less aligned than this one? Is a very personal sort of statement. And then sort of the last one are sort of these dynamic experiments. I don't know why that video didn't load. Where you have sort of this example where we have a flowing foam. So this comes up quite frequently in sort of food science or chemistry, where you're looking at sort of cellular structures, and you're able to get these really cool images of a foam flowing. And so you can look at it and maybe generally tell it looks like the foam is flowing upward, but that's not a very scientific statement. You want to be able to say, is the foam moving quicker in the middle or near the edges? Do larger bubbles move faster than smaller bubbles? How does this compare when you use different sort of, more or less viscous agents or mixtures in order to make this foam? And those are all the things you're interested in quantifying. And from just these images, it's very, very difficult to do that. And so clearly this has not only the problem that you have to analyze each image, but that you have to look at sort of a time series and come up with a way of putting all that information together in sort of a reasonable manner. And so there's lots and lots of examples where that gets very, very difficult and it becomes quite challenging to try to quantify anything well. The last one is, of course, one of the most difficult things of, is the topology of the bubbles changing? Are the neighbors of each bubble shifting as they're flowing? Because if they're shifting, that changes the mechanical properties of the flow quite a bit. The other thing that we sort of will get into a little bit with this course, obviously not too deep, but just sort of an awareness for these problems, is that sort of computing has changed. And so I guess, has everyone heard of Moore's law before? Yeah. Hopefully the computer science student is comfortable with that. So basically this law says the number of transistors doubles every 18 months or so. And so this was really the basis of sort of everything that happened on computers for a long period of time, where, as computers were progressing, the number of transistors were doubling and the clock speed was also going up. So the clock speed was never part of it, but it typically increased as well. And so what we had was, for a long period of time, if your problem was too difficult to solve, you could just wait a year or two and computers would get so much faster that they would just kind of solve it for you. And so you didn't have to come up with very clever ways to solve problems because computers just got better and better and better. And really what started to happen, particularly with speed, is that that's peaked. So that computers aren't getting any faster. Computers 4 years ago, in terms of how many operations they can do per second are still the same as they are today, but there's more transistors available. And even this number has started to flatten off a little bit as well. And so what this means is that you can't simply rely on more processing power to magically solve your problem. And so really what this means is you have to come up with new ways of solving it. And so now, I guess for most of you, if you have a newer or relatively new smartphone, it has multiple cores. I think my phone has like 6 cores inside it, which means you have 6 different processors which are capable of doing tasks. And so you have to be able to come up with ways to divide your problem into little pieces in order to get it done faster. So it can't do any of the pieces individually faster, but it's able to do more things at the same time. But this is a very big challenge because a lot of the algorithms, and particularly algorithms that you write yourself, aren't optimized for parallelization. And so how can you make sure your task is able to scale well without having to deal with all of sort of the intrinsic of using a really fine-grained control of what each microprocessor is doing and threads and semaphores and everything else. What also sort of fits into this is this idea of sort of GPUs. So NVIDIA, AMD, Google are all spending a lot of energy developing new kinds of chips, which are in fact much slower than the processors that you have in your computers today, but they're capable of doing a huge number of things at the same time if you structure your problem in the right way. And so if you're doing large matrix operations, you can do them on a GPU very efficiently because you don't need, because every single sort of core is doing exactly the same step. And so a lot of this then becomes about how can you make sure whatever you're doing fits well to the sort of architectures that exist. And if you try to, hand do too many of these operations, then it won't be flexible to go to these other sort of platforms. And then sort of computing has changed a bit in terms of cloud computing. We won't go too much into this, but Amazon, Microsoft, Switch, Google, all of these companies have cloud platforms, and it's become much, much cheaper to rent computers in the cloud than it is to set up a computer for yourself if you're not using it all the time. And so particularly for large data analysis tasks, you can get a computer from Amazon with a terabyte of memory to analyze whatever data set you need and then give it back to Amazon when you don't need it anymore. And if you were to try to buy this computer, it would be very expensive and you'd have to do the updates and install everything and manage it. And with this, you can just borrow computers quite quickly. And so, yeah, particularly if you look at, Switzerland as a starting point where PhD students are 400 times as expensive per hour as computers from Amazon, just as sort of a rough scaling factor, which means that if you spend an extra hour trying to get something working on your computer here, that's 400 hours you could have been using Amazon machines to solve your problem. And so it's a lot about being aware of how much time it takes to manage computer infrastructure and how you can really easily outsource your problems. And so we won't do anything directly with the cloud in this course. I think there's courses that cover that in more detail. But a lot of the things that we're doing with MyBinder, as you see here, which is where the course notes and everything are hosted, is all cloud-based, and Kaggle as well. So has anyone heard of Kaggle before? A few people. Okay. Has anyone taken a class with Kaggle? Because I think one of the machine learning courses, they use competitions as part of the class, and I think we might have a few examples of that. But Kaggle's all cloud-based, and so when you go on to Kaggle and do a notebook there, or if you are running this here, this actually isn't running on my machine. It's running on some borrowed machine in the cloud that I can use for the purposes of this course, and then when it's done, it will get shut down. And so it's giving me that flexibility of having an easy to access computer And if you go to sort of the course website, and we have this top right, Launch Jupyter Lab. That doesn't launch anything on your computer. That launches a cloud instance that has all the course material on it, all the data sets you need, all of the libraries installed, the right versions of all the tools. And so it makes it much easier to reproduce all the results that I have and all the slides because everything is available in sort of a common reference environment. And so, yeah. So now we'll sort of get to more of the course overview, where the first lecture is this sort of introduction and workflows, and we'll finally get into that after all of this. The second lecture is sort of a new lecture this year, so there aren't any slides from last year. And this is really about ground truth, which is sort of how do you build a data set and augment a data set, because that was sort of one of the biggest shortcomings we had in previous years, that particularly as you start to get into more advanced techniques, it's very important to have ground truth in order to know that you're doing something sensible, and that most projects don't come with ground truth included. You have to spend quite a bit of time preparing that and organizing that yourself. And so the second lecture will really go over how to do that well. The third lecture is then sort of image enhancement with Anders Kastner, and he'll cover how do you enhance images, what sort of tools are available, how do you decide which tools to use. Then we'll get into kind of the more segmentation problems, ultimately ending there with supervised problems and segmentation. And so how do you bring different tools together and sort of let your algorithm learn from data rather than hand coding a bunch of rules. And so we won't really cover any deep learning in this class, but we'll show how that works, and if you're interested, give you the tools to play around with it more yourself. But it's more about leading up to that and showing what the standard things that are available are and how you can at least be aware of what you can solve with deep learning, if that is a good fit for the kind of problem you're working on. Then from there, there's sort of a gear switch to these sort of analysis tasks. So once you've come up with the segmentation, so kind of what are you interested in your image? What are the cells in the background, or tumors inside of lungs, or any number of other tasks you might be looking at? How can you quantify what you actually see? So how do you take a cell and make some meaningful measurements of it? Quantify shape, texture, patterns. Then how can you bring all that together in order to analyze lots of cells? So how well aligned are cells? What pattern do they form? Do they sort of cluster? Are they spread out? Then getting a bit more into dynamic experiments. And so how do you take these tools and apply them to a whole time series of data and know that you're getting kind of reasonable results out? And then so the statistics, prediction, and reproducibility. And so once you've done all these analyses, how can you start to draw conclusions from them and analyze them in a reasonable way? Then there'll probably be a few guest lectures. The exact schedule of that isn't fixed yet. And then the kind of scaling up and big data. So all of the lectures will cover how you build things up so that you can scale it later. So it's not like this is the only one that talks about big data, but this one really talks about how do you take the analysis you've done before and easily run it on a whole cluster of machines. And so what's quite nice about this room here is all of these machines are in a cluster of computers. And so you can submit jobs locally and have them run to do complicated analysis over large data sets. And then finally, sort of the last lecture will be the project presentations. And so everyone who chose to do a science project, or each pair or group that chose to do a science project, can then present what they did, what they found, why they did it, and hopefully get some insights and interesting discussions about how to apply these tools to real problems. Yep. Okay, so I think we'll take a short break here, and then start with sort of the actual material of the course.